And basically what we're doing today is we're going to be spending some time looking at uses for Blast. And I think that you'll find that Blast is actually quite an amazing, I would say quite an amazing piece of software. Just the whole idea that you can, for instance, that you can search through seemingly all known sequences you can take any sequence you want and search through everything that's all known sequences. Is just baffling. The whole idea that that's even possible is just, I mean, just extraordinary. I mean, imagine, for instance, going back in time and telling people that you could search the world, literally, for any piece of, any string, any piece of sequence information. And I think that you'll find that that is just, in itself, just extraordinary. So let's go ahead and transition to the, I don't know why you can't see it on the thing here. Let's go ahead and fix that. I have to pin this. There we go. Hopefully everyone can see this on the screen. But these are the slides that we're working with today. We've got some, we're going to be looking at the website for Blast. We're going to be spending some time actually writing some code. Hopefully that code will work out in good time. I do find that sometimes when you're working with code in bioinformatics, one of the things that seems to always happen is that there is some kind of an error that shows up, which is really kind of annoying. But I'm hoping that we can get over that. So let me just go ahead and get myself sorted, and we can start. Let's see, where's my terminal? I'll need that in a second. There we go. Right. Okay, so let's go back to the slides here. And you'll notice that, for instance, what we're doing is, what we did last time is we began this whole kind of discussion over how Blast works. And so I wanted to spend some time today just to give you some kind of an overview again of what Blast does, and how it looks, and how you use it. That's just so that we, I mean, you'll be using this probably again and again and again in your career in bioinformatics, or any kind of research that you do in bioinformatics. I think that Blast is a very common piece of analysis. So basically, what we have here is I'll just take you through the thing here. So when you start up Blast, I'll just take you to one of these slides. Well, we'll go through the slides first, and then I'll actually work you through an example. But when you start up a Blast thing here, you have to first of all find your sequence. That means you have to find out what it is that what sequence you want to look you're looking to search for. And by that I mean you have to find some kind of a protein sequence or a snippet of DNA or something that you're looking at, and you would like to see whether that protein sequence is known anywhere in any of the databases that we have, or whether that sequence of DNA is known across any genome that's available to science. In other words, I find a gene, or somebody gives me a gene, they don't know what this thing does, and they say, what does this gene do? Well, by looking at the code, I couldn't tell you what it does. And so what I have to do is I have to take that sequence, the gene sequence, the DNA sequence of that gene, and I put that sequence into Blast, and then Blast will then look around it'll compare that sequence with all the other pieces of known information, and it will say, I found a match. I found that this gene that you found, or this sequence you found, is actually a gene which controls some kind of receptor on a cell or something. It makes a protein for a receptor on a cell. And I would only know that if I had been able to align that sequence with every other sequence which is known. So you start with a sequence, you find your sequence, and we'll go through all these steps in a second here, but just bear with me. The next thing you need to do is in Blast, you're going to be entering this accession number, or it's like your, in database speak, that's your, what you call your primary key. Every sequence, every piece of cataloged genetic code in the Blast database, or an NCBI, has its own primary key, which basically means it has its own unique identifier. And your identifier is going to look like this, where you have NC_045512.2. That is the special number which is given to, in this case, I'm looking at the virus which was recovered in Wuhan, the coronavirus 2 isolate complete genome. Remember that I would like to say that this virus was discovered in China by Chinese scientists. We have no idea whether this is the proverbial China virus. It was discovered there. I think it's very important that we use the correct language when we discuss results, because in the if you look at all the hatred which is going on for Asians in this country, I think that perhaps if we cleaned up our language and we were more specific about what we're actually talking about, then this miscommunication would not perhaps occur, which would incite violence. And so we must be clear when we say that these viruses, the viruses that we discussed today and the viruses that we had over the lab, those were the influenza viruses. Those viruses were discovered in Asian countries, but it doesn't mean that they were actually originating there, or they were, we don't know that. So this is the virus, the coronavirus, which was discovered in Wuhan. And so that's how I'm going to be addressing all of these sequences. So we have so anyway, once we have these sequences, we put the sequences into the BLAST parameters base, which is right here. You're basically putting in a query sequence, and then again, you're basically putting in this number here, which if you look at the locus, hopefully you can see what I'm looking at now, but it's like this number, NC45512, that's what they call the locus number. It's also the same number as the accession number, which you can see right here. Now, those numbers, again, are the special, I guess, the variables you'd use to describe these sequences in a database. And so you put those variables right here. One thing I will say, though, is that this 0.2 that we see, this 0.2, that's, there's the accession number plus 0.2. This number, you can see it's the accession number and the version number are the same. That 0.2 is the version, which means, this is actually a very interesting topic, that, for instance, the COVID genome was first of all found, it was sequenced. That means they were actually able to take the biology and then determine what the genetic code is from that. And once they had that code, then they were able to say this is version 1 of the code. Now, the reason why they have 0.2 is because this is the second version of this particular virus. Now, you're probably thinking, why would they have 2 versions of this? I mean, isn't the first time good enough? Well, actually, errors do creep in when you're actually creating your code, when you're actually determining what the string is that is DNA. And so when those errors are found, then they change the version number, and then they put the latest version into NCBI. But you can still find the original versions of them. They're still there. Nothing ever gets erased. It's all still there. But you have to kind of look for the most recent version number, because that's the version that's going to be the one that hopefully science will use. But this actually makes a problem if you think about it, because when you change the version of the genetic code that everyone's, or that the analysis is using, when you change that number, that means that you may have to go back and change some of the analyses which have used that original version. So for instance, if you ran an analysis in computer science using a library, and you find out that library has changed, or somebody found a major bug, so that the results that come out of that library are not, they're no good, then you may have to go back and rerun that library and run your analysis all over again. And that can happen. And that can happen in bioinformatics too. And so as technology becomes better and better, the libraries in software become, they have improvements, and so sometimes you have to go back and rerun some of your analyses if the library has changed, because the library may have been found to have a virus, or not a virus, a bug. And the data also can sometimes change because it's become improved or more sensitive or better data or more realistic, or somehow the technology has been to make better data. And so in that case, you might consider going back and rerunning some of your data. But in some cases, though, it's just not possible. You just can't go back and do that. And so it's really up to you, the bioinformatician, to decide for yourself and for your lab that this is an important piece of maybe an important piece of data. Maybe you need to go back and check to make sure that it's working with or that your analysis is correct with the more recent. Again, that's a decision that only you can make. Anyway, but once you put that number in here, then you have to put some other options. I just made a screen copy of all of these things just so you have an idea about what the general options should be. Typically, in the program selection, you'll see down at the bottom here, I typically put down optimize for highly similar sequences. But you can also optimize this thing for somewhat similar sequences. And what that means is that it's how extensive the search is going to be. You might find, for instance, that some searches, I'll just say hypothetically here, but some searches might take a long time to run, especially if you have like a really large string of code that you're, or of DNA or of protein code that you're trying to find a match for. And so maybe that'll take a long time. And so you can change your optimization for option for highly similar sequences, or dissimilar sequences, or somewhat similar sequences. And you can see how those results compare to, to the actual results you're looking for. Anyway, once you're finished, you go ahead and push the old blast button, which is right down here. And one thing I think you can do, maybe I think that somewhere, well, on some renditions of Blast, some different websites have Blast code on them, and so you can run code over the different. I'm saying the different websites that have sequence information. You can run their version of Blast, and sometimes on Blast you can actually give them. You can put down your email so that you don't have to actually wait around for the Blast to finish working, but rather they'll just send you an email of the results. I've seen that, but I don't see that option here. It must be here someplace. There must be that way of doing things. But anyway, but still, you basically wait around, and so this will. You can see that this could take some time. In fact, I've been actually waited for up to maybe 10, 15 minutes in some instances, actually to get my results using this website. And so you just kind of leave the web page open, don't close it, but just leave it there, and then eventually something will happen. And if your results come back, then you'll see them here, but it'll say not that it'll say no hits. You're basically always bound to find some hits. But you might find that it would say something like, there's been an error, too many results, or maybe too few results, or something. I mean, maybe it'll tell you whatever it is, and then you may have to go back and adjust some of these parameters there. Once you get your results, though, you're going to have a page that looks like this. And I would say that this is probably the most important page. This is really, there's graphics and other stuff that we discussed last time, which we'll talk about briefly here. But this is the most important page. And primarily, it's important because you have these columns here in the red box. You can see that you have some information about the sequences that come up in the results. So what are these sequences that come up in the result? Well, these sequences are the sequences which were in the database, which Blast found to resemble the sequence that you sent up here. It just happens to be that the sequence that you put up here was the virus isolated at Wuhan. And it was the complete genome, but you find, for instance, that other sequences which are very similar to that have also been found in the results. And when you look at the title or the descriptions of these sequences, you find out that they are the SARS-CoV-2 virus, coronavirus. And so that's really how you can tell that what you're looking at now is the virus, because otherwise you may not know. I mean, in this case, let me just go back here. In this case, we were lucky that we knew what this thing was. But if we didn't have this title to tell us what it was, then we could go into this enter query sequence and just dump in the sequence there, and then ask Blast to tell us what it finds, and that's where all this information here would come from. But actually, this analysis that we're doing here, we knew what the sequence was going into the analysis, and we were actually asking, what other sequences are out there which is similar to this sequence that we're looking at? And so these are the other sequences which are similar. These sequences are variants. That means that they have been isolated from patients we have to treat this data with respect, because there is loss of life probably involved with each one of these sequences here, and human suffering. And so we have to treat these results with respect. But we realize, though, that each of these sequences, though, was isolated from somebody who had COVID, or they found it from some other source. But it's still, they knew at the time that it was COVID. They put this sequence into the database saying this is COVID, and then our search uncovered these sequences and said that, and found out that the sequence that we were using, or the one that we were searching, was actually very, very similar to these other sequences. And so if we were the bioinformatician here, we'd say, these viruses are COVID, therefore the sequence that I was originally working with must be some relation of COVID. Now let's just talk about these scores for a second here. In fact, on the next page, here we go. On the next page, I have some sense of how you can understand the stuff that actually shows up in this red box. And that is we have the max score. I'll just read this. It's the score of the best matching segment for local alignment, not global. So it's like you're looking at like chunks of code inside, like subsequences of code inside a sequence and comparing those. I never actually bothered looking at the max score, frankly. I don't bother with that. What I do look at, though, is some of the other ones, and we'll talk about that in a second. Then we have the total score. The total score this basically scores all matching segments found as some max score if there is only one segment. Okay, that's important. Some research analyses require that you know this max score just to add some extra information to your analysis. We have the query coverage. This is the percent of the query sequence that align to some part of the match. That's actually getting more important here. So the query coverage, 100%. If you see on the very, very top line, I'm looking at oops, hopefully I'm not going to crash my computer by zooming in too far. But you can see that this query coverage here, 100%, 100%, 100%, 99%, 100, and so on. As you get down the list, you'll see that the numbers will drop. But what that's saying, though, is it's looking at the code that you've just put in there, and it's finding out how much of this code that you're querying is the same as the code that's in these sequences here. Well, in this case, because it's almost exactly the same sequence, it's saying that almost the whole sequence is the same. But if you find like a lower number, it's saying that you're finding maybe chunks of the code which are inside the sequence that we're aligning, and those chunks are are similar. So if you find like 50% query coverage, it means that maybe half of the sequence is different and the other half is composed of chunks or subsequences which are the same between the sequence that you're working with and the found sequences or the sequences in the result. Now, the most important number that I look at, and maybe some other researchers too would use this one more often, but it's the e-value. And the e-value is actually kind of interesting here. All of these numbers, by the way, they all have something to do with how similar something is. But only the e-value has some kind of statistics in it, which is saying, I'm getting a similarity, but statistically it's virtually impossible to have this similarity happen by random chance. That means that there must have been something to make this sequence into this sequence. There must have been some reason why the sequences were the same. So it's saying it's statistically impossible for this to be so similar randomly. It's a statistical measure evaluating how likely it is that a good match could occur by chance. And that's actually an important note here, because if you think about it, when you're using BLAST, you're looking at, terabytes upon terabytes upon terabytes of information. You're comparing your sequence to this wealth of information, like all sequences ever that are in this database. And the database is huge. It's like a giant server farm that's running just the database. And so it's very likely that you're going to find your sequence, whatever it may be that you type in, it's very likely that you're going to find this in a number of different places, but there's no direct connection between the sequence or the sequence that you put in there and the sequence that you find. It could just be simply random. Even if you go out for like 20 different bases or 40 different bases, and you're just randomly pushing A's, T's, G's, and C's, let's say you're just typing in a random sequence in this enter session number or the FAFTA sequence. If you're just typing random code in here, and you can try this, you're probably bound to find something. And that something that you find is very likely to be random. That means that it just happens to be that whatever it is you typed that day with on your keyboard just happened to be found in another organism. And when you're looking at the wealth of information that's out there in this database, it's very likely that you'll find at least one hit. But this e-value, that is where it says, yes, you found a hit, but it's very unlikely that it was random. It must have been somehow left over from some evolutionary pressure, or maybe there was horizontal gene transfer, maybe it was a series of mutations over time, but maybe there was some reason why this sequence is actually the same sequence. It's like it's saying that it's not a fluke, it's not random here, but there is something there to suggest that this was evolution in action. So that's your e-value, and that's a very important value to look at. So many researchers will report the e-value when they use BLAST. They'll report the e-value first, and then they'll talk about some of these other scores too, just in case that's the way that you do your analysis. But for me, though, my analysis is the e-value. And then we have, for instance, actually I'll tell you something else here. So lower E scores indicate that both sequences are truly similar and are not similar by chance alone. Identical sequences have scores of 0. So if you have an E-value, which is a high E-value, higher scores are not good scores. You want low scores. Low scores means I'm really, really that these sequences have some common thread. It's not random. Low scores will say it's not random. High scores say that there's more randomness, creeping into the sequences you're looking at. So maybe the sequence that was returned in your result, it's a random, which is that sequence is just randomly there. It doesn't mean there's any evolutionary connection. Then we have the max indent. This is the percentage of nucleotides that are identical between the query and the target sequences within the matching regions. And so, for instance, here, if you're looking at these sequences, you'll see it's like your identification, percent identification, or the percent identified, I guess. But you're saying that 100% of the sequence, base per base, is the same, base per base. And so that's interesting. I mean, you're actually seeing that the scores of these sequences are very, very similar. Let me just see something else. Can we see from here? No, I was going to say, maybe, for instance, there's some more information that you could use inside some of these. No, never mind. I was going to say that, for instance, you might be able to see sometimes on these records, you see some pieces of information about, they might add some information about some of these percentages from like papers that, like some research papers that you might find, just report on these percentages. They just do that. And so sometimes I was looking to see whether, if I had enough information in my record here to see some of these papers, and sometimes you don't. But sometimes, though, when you look at your gene bank record, you'll find some papers which would say, yes, we've discovered this complete genome of this organism, and in the title you'll see something about how it says, although we didn't know at the time what it was, we know now from our BLAST analysis that this is, whatever the organism may be. Now, so then you have that, and then you have this accession length here. You have the length of the sequence itself. In fact, let me just go back. I had to make you feel like you're being sick there. I'm looking at the length. Now here we have, for instance, let me just show you something else. So in this complete genome that we have for the virus, which was discovered in Wuhan, this number right here, 29,903 base pairs of this RNA. 29,903, that's your RNA code. Now, if you go down here, back to the results, and I do apologize for making everyone feel woozy over all this flopping around business, but you'll notice that these viruses, too, that we see in the list, have similar lengths. So this is your length right here, 29,000. This is this list, or this virus at the very, very top of the list is a different virus, but it is still 29,903. And here's another virus, which is 29,904. And then there's another virus down here, 29,901. Let me just ask the class here, what do you think is going on here? Why are the lengths of these particular sequence results different? Why are the lengths different? What would you suggest? If someone asks you, what would you suggest? But listen, ask you, Catherine, yes. Maybe there's some kind of I think we have it here. It's maybe a mutation of some sort. Maybe it's an insertion, or maybe it's a deletion. And so the thing is, though, that we're likely to look at our. So let me just say something else here. We're likely to go back to our original sequence and say, aha, we are 29,903 base pairs. Okay, so that's what I'm working with. So that is my sample here. So everything is 29,903. And so then if I find a sequence like this, which is 29,904, I'm likely to think that my sequence is the template, and everything that I find after that, that is the sequence that has the mutation. The 29,904 is a 29,901, but that's actually a misconception. We don't know that. Maybe the sequence that I'm actually comparing to the database, in other words, the sequence that I began with, and maybe this isn't the best case, to show, but I mean, you might find, but this particular sequence here, this one here, maybe this is the one that actually received the mutation, that actually received the insertion or the deletions or whatever it may be. And for all I know, getting back to my results, I don't know, for instance, that perhaps this is the second sequence on my list, or the third, or the fourth, or actually, how about this one, the 29,901, this guy here, if I can get down, if I can zoom in without crashing my machine. But I don't know that maybe that isn't the original sequence. And the sequence that I'm actually working with actually sustained 2 insertions, or 4, or 3, actually it's 3 insertions, I guess, wouldn't it? 2 insertions. So I don't know actually whether, you just don't know these things. And so the reason why I'm saying this is because many papers will come out saying, we have the sequence. We found this sequence in this organism. We did a blast search and we found these other sequences. These other sequences are therefore distant relations of our sequence. But you don't know actually whether maybe that's the wrong language, because maybe, for instance, those other sequences, your sequence is actually, for instance, the sequence that drifted away evolutionarily to the other sequences which you find in BLAST. And so you have to be aware of that, because when you're describing your results, you don't want to say that the sequence that you have is the template or is the only one, but you have to just say that there is a distance between the sequences which I find in my database and the sequences, or the sequence that I'm actually holding in my hand. But you don't know which one drifted first. You just know that they're not the same. And that's actually a problem, because you'll notice when you do your research projects, there are some scientists who are doing their research and they're talking about BLAST results, and they're just assuming that the sequence that they're holding in their hand was the first one, and everything else that's in the database was the second, third, fourth, fifth, and so on. And so it may not necessarily be the root of the tree that you're trying to build. Okay, so that was a rant, I'm sorry about that. And speaking of trees, you have this phylogenetic tree to show types of relatedness. And so you can kind of get an idea about where your sequence is in relation to the other ones. These are still the viruses, or the virus which was recovered in Wuhan. And you can, in fact, I've outlined it at the bottom. But you can see, though, that according to this database, some of these sequences are similar, but they're not, they're not the same. And so you can see that in this tree that there are variants to the particular sequence. So yes, there's a lot there. So BLAST is cool. Let's just try an example here. So we do have some code today, but I think that maybe the website might be more fun for a second here. But if you're trying to start BLAST, in fact, I think that I actually have a link out here on my slides. Is that right? Well, we'll get to the code in a second here. But you do have this really cool BLAST here. In fact, this is a cool horse. Why not? To find BLAST, you want to go to NCBI. What I do is I just do a search for NCBI, but actually the website is ncbinlm.nih.gov, and hopefully you can see that from your deal. And when you type in all this information here, you'll find, for instance, that this is what the website looks like. And so I'm going to go and just do a search for something. I'm going to type in, let's just do our own analysis. I'm going to type in a protein that's actually fairly commonly used across many organisms. It's called the SIRT1, SIRT1. You type that in at the top here where it says, you can see that there's like all databases. I just use that. But you type in SIRT1 at the top, and hopefully you can all see that. Press enter. And SIRT1 will then show you all the results for SIRT1 will show up. And you have like this nice page here, which is relatively new. They didn't have this years ago. But it kind of like gives you some ideas about what this is. You can click on this and get some more information. But actually, though, I'm not going to do that right now. Go down to this area here, which is called nucleotide. Now, nucleotide is actually very important because we are working with the DNA sequence, and so we need to find the nucleotides, and that's how you would find the sequences for SIRT1. So if you click on nucleotide, you'll notice that your database up here has now changed, so you're not looking at all databases, but rather you're looking at nucleotide. But if you go down here, you'll find all these different hits of SIRT1 in the literature, or not in the literature, but in the sequence data that they have. I should say something though, and that is that the search engine is perfect. It's not a perfect search engine, because in some cases you find, for instance, this first reference, which is spot on. That's exactly what cert1 is that we're looking for. That's the sequence we're looking for. But in other areas, though, you'll find, for instance, like number 4, for instance, that's not exactly what I'm looking for here. In fact, it's not even the word cert1 isn't even in the title. And so you do have to be cognizant of these things. If you're looking for cert1, I would say spend some time to look through all these different records and make sure that it actually is the particular sequence that you're looking for. Another thing you can do, though, for instance, the sequence that I'm looking for is basically around, yeah, here we are, 375 base pairs. I was thinking it's like 360, but it's about that. It's about that. So the sequence that I'm looking at. And you can find, for instance, that this sequence here, for number 4, is, well, first of all, this is DNA, and I'm looking at messenger RNA, but it's 2 million bases. And so that should tell you by the size that it's probably not the sequence that you're looking at. So I'll go ahead and click on this cert1 over here, and I get this record there. Now, you'll find that the record has a lot of information there. You have information about, for instance, the accession number. That's what we were working with before. This is the version number, where you have the accession number plus this .1, which means that this is the first thing. It's been the first rendition of the sequence. So it must have been that since 14, when it was first sequenced, that since from 14 to today, nobody has found any updates to the code. And so it's, for this particular organism or this particular strand, it appears to be this is the most recent version of data that you have. Then you have information here about the organism, where it's come from. You have this is something about the family tree. You have some the authors that stand by their work to say that we isolated this particular sequence of genes from this organism, and this is the sequence. So these are the authors, I would believe, who either put the sequence in this database or had something to do with actually determining what that sequence was. And then you have the reference, so you can actually go and find this journal and find out, what more the authors had to say about this thing. Then you have some other information here too. In fact, for instance, one thing that's kind of interesting is you'll find that there's like authors and information seemingly throughout the entire sequence, and that is because the sequence information just the DNA or the RNA, but rather there are chunks of code inside here which are talking about the translation, which means there's actually chunks of code or genes inside the sequence which create a protein. And so that is the protein which is actually created by this piece of code. Somewhere in this code, embedded in this code. In fact, in somewhere, this is saying that from base 1 to base 375, which I believe is the entire thing. Yeah, the whole thing is 375. So we're saying that this gene, this is a gene here. This gene, when you translate it, you get this protein code there, and this is your actual code you get here. That's your, what would that be? Your RNA, I think it is. Yep, that's your RNA, and it says it's RNA right at the top. Another thing you should look at, though, is you have the length of the thing. You know that this is RNA, but also you know this is linear. That means it's like the ladder. It's the twisted ladder, the helix. A plasmid, it would be kind of a circular piece of data of genetic code here. And so if you're looking at, for instance, a bacterium, you'd have like a round kind of, I guess, a round circle of DNA. And that's how bacterial DNA sometimes comes. This is linear. And another thing that's interesting about these names of the authors here is Fernandez and, I guess, his team or her team. These were the ones who isolated this particular piece of protein, and they may have done some other studies on this protein to determine some of its properties. And so that's really the record that goes into NCBI. And so when you're looking up a gene or when you're looking up a protein, do a search and look for the nucleotide of record of that gene, and you can find all kinds of information about the origins of this sequence, where it came from. In fact, where it came from would be probably mentioned in here. But also, like, how that this sequence is a gene, or how do they know that the sequence is a landmark for something else? How do they know that? Well, you can find all these so-called landmarks, or the proteins that they create, and you can read up on those papers here and find out how they know. Anyway, but that's all very interesting. So that's your kind of your overview of some of these records. We're going to go ahead and click on this Run Blast. You can see over here and analyze this sequence. I'm just going to go ahead and click on Blast. And you'll notice that when I click on Blast, I get exactly the same page that we saw in our notes. And the accession number has already been entered in, very kindly. And I don't touch anything here. I just leave everything absolutely, as the default values. And you just need to hit the Blast button. Let it go and see what happens. Now, this is only number 375. So it's not a very long piece of code here. And so the Blast is probably not going to take a long time to do. But if you're working with like a sequence that's like maybe millions or even like maybe 20 million base pairs long, and you find those, then your Blast could take some time. So that's going to be a thing. And so but anyway, these are the results, and so I can look at these things and say, okay, here is the Cyprinus carpio. I think that's a type of a fish. You can find that the e-value is 0.0. And so remember what the e-value tells us. It's saying that this is not random, that other organism has the same code in there, probably from some kind of evolutionary pressure. And since this is 0.0, it's suggestive that there was a common relationship. There's a common relationship between this sequence on the first sequence here and the sequence that I was actually doing research on. The sequences, the e-values get close to 0. This value over here is just saying that this is not 0, but it's very close to 0, and that's good enough. But you can also see in this number right here, the percent identity, which has 96.53%. That suggests that there is some evolutionary distance. The e-value is so low, though, that evolutionary distance is probably indeed evolution, but there are chunks of code inside the sequence which are not the same. It's like there's some mismatches. And so you can kind of like, as you move down the list, you'll see that the e-values grow bigger and the percentage identity gets smaller. And so, for instance, you find that this is like 82.76%, which is suggesting that there's chunks of code that are different. Let me just go ahead and just click on this. If I go back over here, I think I can just go ahead and do this. I can click on the name, and there you go. So when I click on the name, we've seen these types of arrangements before. This is the sequence by sequence aligning it, or the alignments of sequences. This is what we saw when we worked with the Needleman-Wunsch algorithm. But you can see that there are certain areas, like right here and like right here, where there's no line between the bases. So that says that right about here, there's a C, and right about here, I see a T. That means that my sequence is different from their sequence. And so perhaps this suggests that there's a mutation in my sequence where maybe my C was somehow, I don't know, it sustained some kind of a mutation where originally it was maybe an A, for instance, or maybe a T, or actually maybe it was a T, and it became a C. So maybe the T was bumped out and replaced eventually with a C. And so that could be an indication that there was some kind of an error that was made, and it was incorrectly fixed. Over here, for instance, we see an A over here, and then we have a C down, or a C above. Again, maybe the C was an A, and you find that that A was replaced with a C. It would be an interesting research project to go through viral DNA code and find out whether, when you find a mutation like this, whether the A's, or whether you find usually that there's a mutation between or a mismatch rather, are there more mismatches between C's and A's, or C's and T's, or I mean, was it that, for instance, there was an A originally right here, which was somehow copied incorrectly, it became a C, and it wasn't corrected? Was that a problem that happened here because there was originally an A? Maybe the A wasn't wasn't held together very strong in that code. Whereas if you have like a G, maybe you're less likely to have a G mutation because G's have 3 bonds, and therefore they'd be more likely not to get bumped out by mistake from whatever reason. Anyway, but research into why the mutation happens, that would be actually very interesting. But you can see, though, that there are gaps. There are not gaps necessarily in this, but there are mismatches. And here's a gap. Here's a gap. Right down here at the bottom, we can see that dash. That's a gap. Are there any gaps here? Yeah, here's a gap. Here we are. There's a gap here, and so that's suggesting that, for instance, maybe this A here was either inserted in the above sequence, or maybe something was removed, deleted, and it was never put back. And so anyway, that's another piece of information that you can study if you wanted to find out, like, how do we explain these mutations. But as we know from our studies in like cystic fibrosis, just having this particular tiny mutation right here does have catastrophic outcomes, and that is that people who have that mutation at a specific, like one mutation at a specific place, do have cystic fibrosis. Just one mutation. It's just extraordinary. Anyway, you can go back up to the top of this, and you can kind of you can see the other sequences. All the results are there. It will take you some time to climb up to the top because there's just so much data there. So I'm just going up to the top here. But I think that you kind of get the idea about how you can use BLAST. And so I'm going to go back now to something else. I was doing some research on using BioPython. I thought, it takes, sometimes when you're using BLAST, to work with a database. And so I was looking around on BioPython to see whether BioPython has a solution that allows us to do BLAST, like, just run it in an automated sense where we're just like kind of letting it go. You leave your computer running at night, and in the morning you find that all of your BLAST results are waiting for you. And so I came across this really good, in fact, I'll put this out there if you want to see this. I'll put the chat out here, just so you have it right now to look at. You should find this in the class chat. But it's actually quite a good reference. And what this reference is it's basically kind of like your instructions for how to use BioPython for seemingly all of the analyses that it does. And so you can do a whole wealth of things. In fact, there's ClustalW, which exists as an alignment tool, as we saw. But we have BLAST also as an alignment tool that you can run in BioPython. And so looking through the pages here, you find that there's this one line. Where is that? Here, command line. It's this one, I guess. It's right here. This is like your actual code. And so I've given you some code in class, and I wanted just to add this line and actually fix up the code and to see whether we can't get this this thing working, the code that I gave you working here, to see whether, just to kind of get it working. But I will say this, though, that when you're working with online tools for where you have databases involved, like if you're working with BioPython to work with BLAST, if you're writing a BioPython program to interact with BLAST, one of the problems that I found, which I found many times, in fact, not just for like last night, but I find that it's sometimes difficult to actually get results in a timely way because the servers that are serving your BLAST client can crash, or maybe something happens in the middle where the connection is reset for some reason. So in other words, what I'm saying is that when you're using this BioPython BLAST client, which we're going to work on now, you'll find that, for instance, it doesn't always work out. It doesn't always work perfectly. You might find that there are some crashes. Let me just go ahead and get my my notes up on my screen here, and we'll start this momentarily. But I'm going to go ahead and find a piece of code in here it's the BP BLAST _ class, and we're going to try and modify that to see whether we can get some. Whether we can get it to work today. So let me just get to my notes, and I will get you there. Where are we now? All right, I have to get my sandbox. It should all be there. All right, so I got that up on my screen. Now, okay, so, by the way, there's an activity after this, so please don't forget about that. I'll put the activity out in a second. But I'm going to go back now to, if I can find my prompt, browser, terminal, editor. I'm going to load up my editor with the code that we're using in class. Let me just go ahead and show you what I'm going to do. Hang on, let me just get my editor working here. For some reason I'm not seeing it on my screen. Where are you, editor? Okay, so, oops, I don't know why you're not seeing that. Oh, you know what? There you are. And I have to close down my, there we are. Now you should see it. Good. Okay, so now I'm going to go with my editor. I use my editor to actually do my code. I'm going to go, and you can't see what I'm doing here, but actually I'm opening up the file that I have in class. Let me just go ahead and get this. I don't know why this is so difficult today. Is it Friday, the end of the week? We're all just exhausted. Blast. Okay, so I'm going to open up my project that I have here. I'm going to go to my sandbox, open up my class, there, and we'll see what happens. Now, one thing that we're going to need, I'll just give you an idea about what to expect. How do I get back to my thing here? Okay, there we go. Let me go back to my terminal, and we're going to, let me just show you, seeing when this thing runs, and hopefully it'll work, but I'm not quite sure. I'm going to be running actually in this. I'm not using. I use Dropbox. I'm not using the repository, but I'm going to be working with this file in class. But let me just go ahead and show you this. Get into your Docker container, and for me, what I do is I have my Docker container parked in source, src, docker bio, and then I can just go ahead and run this. Whoops, Mac. One thing I have to do is I have to type in sh. Go ahead and just get into my Docker container. Everyone should be able to see where I am. And once I'm inside, in fact, the screen. How bizarre. You can see two of me. That's weird. I don't know why you're seeing two of this. There's two cameras open of me. How exciting. Let me just go ahead and minimize one of these things here. There we go. We should only have one now. And so what I'm going to do now is I'm going to go back to this. I'm going to go and try and run this from my Docker container. And I'll just give you an idea about what to expect. And if we don't get this thing done today, I'll just give you my code so that you have that to work with. But basically, though, I'm inside my container. You can see that this is root. I type in Python 3 followed by BP Blast. And if I just type in py, it's going to be running. What you'll see here is, and hopefully you can all see this, it's not running off the page, but it says enter the version number. Now, version number. And by that, what I'm actually talking about is if I go back to if I go back to my slides, let me just show you something. I'm trying to get my slides open here. If I go back here, you'll find that, let's just go and grab any old version. Oops. I'll just grab something here. Let's go back to get the sequence we were just working on. If I can find that certain one. Hang on. Let me pull that up here. NCBI. I'll do a cert 1. Oops. And I'm going to find my nucleotide. Oops, it's not there because I have this funny character. There we go. I was working with. If I go ahead and do this, click on this record, get this thing up, I'm going to go after this, what I call the version number, this, and hopefully this will do it. Now, one of the problems with working with this particular code is that it takes about 5 minutes sometimes to run an analysis using BioPython. And so I'm going to see how long this thing takes to do. And so I'll let it sit here for a second here while we do some other stuff. But I imagine that, for instance, it's not going to finish in any good time because I tried running it this morning and it still took some time to do. Anyway, but according to the, I mean, if you look at the technical documents here that I'm just looking at, according to this document here, that's the code. What I'm asking is that if you find, for instance, that there's a bug in the code or something like that, or you find a better way of doing something, please do not hesitate to put those in, to make that change, and then to let me know what the change is. I would love to see what it is. And go ahead and just cancel this because it's going to take too long to run, I think. But it should be actually making a call to test right now. If you run this over the weekend, it might work a little bit more quickly. But I mean, this morning though, I had it running for like half an hour and nothing. So I'm just going to use this opportunity just to work with the code. But you get the idea. But what happens now is that when you go ahead and when you get your results, you should have a file which is like sent out. In fact, this is your, I don't know why this file is impartial here, but you should have a file that comes out, which is like this. This is a previous result that I did. And so I'm giving you a parser. Let me just go ahead and show you what this file looks like if you run this. But you'll see that the file is an XML document. It's basically a document which is, it's a vehicle for data. The data is all contained in these little environments here so that you can kind of keep track of what the information is your analysis. But what you get though is you have your analysis, like your alignments, and you have some information about like the e-scores and all these other things. It's all right here in your document. So it's a little bit difficult to read, and so I'm giving you a parser. If you just go ahead and type in Python parser, and I type in the name of the file, and the file that I'm looking at is this KZ501822-1 XML. If you go ahead and push enter there, there's no such file. I'm just going to go ahead and copy this. I probably made a mistake in this. This makes me lazy. And I do this, I'll paste that in there. There we go. If you run this command here, you can see at the bottom, you'll see that you actually get the information about your alignments, which you'd expect to find if this were this information on Blast. So you should get the same. You should get the same information, but this is a different way of looking at it's a different way of actually getting that information. This is what I'd call more automated. Okay, so that's all very cool. Go ahead and start with our editor. Let me just go ahead and get the browser off, get back to the editor, and let's see what we can do. So over in this thing here, if you look through the code, I just want to spend some time. And by the way, if we don't get everything done, I'll just send you my code so that you have that to work with, and you can modify that. If you find a better way of writing the code, please let me know. I mean, in many cases, believe me, your code is going to be much better than mine. That is a fact. That is a fact. Okay, so let me just go through it. So we have in the code here, you have all these definitions here. These definitions are the functions themselves as they are being as they're being entered into memory. You have all the functions basically dumped into memory here. But they're not actually being run just yet, though. All these functions are going in there. In fact, I'll just start at the top here. This is this is all the stuff that's being dumped into memory. This over here is it's a Unix thing that helps you to run this code at the prompt if you wanted to, but I don't worry about that just yet. This is I just dumped this in here anyway in my code, because sometimes in pieces of my code, I'll have like print statements that reference the date or the version or the author mail. You want to have some information in your code also about where the inspiration came from, maybe who wrote the code, all that kind of thing here. And in my comment here, I have information about what this code does, and I have a link about where I got the information. This is just information I use to change the color of the text. I find that working with color is very, very handy. And so this is a function that does parse results. It basically just helps you to get the information back from Blast, and then to kind of show it onto this, take it out of the XML and show it to the screen. This is the version number we're going to be working with here. That is actually getting the information. You can see here, it gets the sequence number for our analysis. And then we have a sequence over here, or a function over here that actually runs Blast, or initiates the Blast analysis. And then we have another function here that saves things. You'll notice in each of my functions, there's like this doc string that kind of gives you a blurb, if you will, about what this thing is doing. That will help you in the future when you have a program that has maybe many different functions. This could help you to to remember what each function is. You'll also notice, though, that the way I code, I'm trying to keep everything very, very simple. It's better having multiple functions than having one function that does multiple things, I think. Because then if a bug happens or something goes wrong, then, for instance, I know if the bug is here, for instance, then all I have to do is go back and modify this function. But if I have a bug, for instance, that shows up in main, then that means I have to go through and basically check a whole bunch of other stuff. Or if I have a really complicated function that's doing a whole bunch of things, a bug could make everything very difficult to fix. So anyway, this is what happens here at the very, very bottom. As you remember, this is just code that makes sure that you're actually running this inside your container. To load the libraries, and if the libraries aren't available, then it says, wait a minute, I'm having an error loading BioPython. Are you actually in the container running this? And then over here you have this main, which is actually trying to run the thing. Actually, that actually runs the code. And main is the driver function. So here it says here, this is what it is. It's Blast analysis. And then we have this version number, which says, it calls to this function here. And so basically it's saying, use this function here, get some information from the user, and whatever the user writes in, send it back to this variable here, and then do some other stuff with that variable later on. And so I think we have to go back and get version number first. And so if we go to get version number, there we go. We're probably not going to get everything done. We have like 2 minutes. I'll see what I can get in here. But basically, your version number is going to be code that's asking the user something. And so your code is going to, I'll just remove this to do. I'll have, like, for instance, a prompt here. I have my prompt over here. And so really what I want to do is I want to have some kind of, I'll just get I'm making a version number variable. And hopefully you can all see this. I can try and make this bigger. Maybe that would be better. Number is equal to input, and then I want to show the user something that I want them to read something, so they have some idea about what they're being prompted for. And then you have this my prompt string. So then, whatever the user types in here, they're shown this, and whatever the user types in that question is saved to this number or this variable there. And then what I want to do is I want to return that value to the calling function. Whoever called this, get that. So here's this. And so anyway, I'm going to go ahead and just do that. So you have this you're saving this. You know what I should do? I should save this with a different name. Sorry. So I'm not overwriting things. Okay, so now that's basically done here. So we have this get version number, and so this thing should work. I get some kind of code or some kind of text that comes back, and there's that. I think that we're out of time here. Are we out of time for this? Shoot, how much time actually do we have? Time flies in this class. I gotta tell you, it flies so fast. Tell you what I'll do. You can look at this and give it and try it out here. I'll wait toward like maybe for an hour or so, and then I'll send out my code, and so you can kind of get an idea about what the code looks like. Just have a look at this and see what you can do. But there is, for instance, going back to I hate to cut this thing short. We'll come back to this on Monday. But if you go back to the slides here, there is an activity. I'll go ahead and put this out right now on Slack so that everyone is aware. But you do have this activity, which is actually quite important to do. It's your it's asking questions over your code. In fact, I think actually tell you what, I'll just send out the code after class.